So let's first of all then define mitosis. And what are the features about mitosis that we have to know? So mitosis is actually just one cell division. You'll see that this contrasts later with meiosis. And it produces genetically identical daughter cells. That also contrasts with meiosis in a minute and will be really clear. So this is my parent cell, and it divides to form two daughter cells. Daughter cells that are also genetically identical. It is used to form somatic cells. Quick reminder of the term somatic. It basically means every cell in the body apart from gametes. So it produces somatic cells and not gametes. And why is that important? Growth and repair of multicellular organisms, as well as asexual reproduction in unicellular eukaryotes. The type of cell division by which prokaryotes divide is called fission, not mitosis. So if you see the word mitosis, mitosis specifically refers to the particular behavior of chromosomes and what the cell does. You will not say that bacteria divide by mitosis. So let's go and see the phases one by one of mitosis. First phase is called prophase. And what are the events that are happening at prophase? First of all, DNA condenses to form visible chromosomes. How does that happen? By DNA supercoiling. So DNA supercoils and it condenses to form visible chromosomes. Prior to that, DNA still exists in the nucleus as chromatin, meaning DNA wrapped around histone proteins. If you haven't done that yet, don't worry, you will. But it's in the form of it just kind of looks like a loose sort of string-like structure in the nucleus, not as visible rods that are chromosomes. The second thing that happens is that the nuclear envelope disintegrates. That's essential if we're going to split the chromosomes. And so here we can see no nuclear envelope. We can see that the chromosomes are condensed, invisible. What's another thing that happens? The centrioles move to the pole. So these here are the centrioles. If you don't remember what they are, revise your chapter on cell structure. Centrioles are those two little organelles that duplicate and they move to opposite poles of the cell at the start of cell division, at the start of mitosis. And the centrioles organize the mitotic fibers. So we'll be able to see the mitotic fibers. Just do them in a different color so you guys can see them better. You'll be able to see the mitotic fibers like that, sort of extending across the nucleus. So the mitotic fibers emanate, we say, from the centrioles. What happens in metaphase? In metaphase is when the chromosomes are actually going to line up along the equator of the cell. And that's when the mitotic fibers or the mitotic spindle, I'm just going to say that it can be called spindle as well instead of fibers. Just be aware of that. Mitotic spindle. Oh, sorry. Spindle fibers. Sorry, my bad. I meant mitotic spindle instead of spindle fibers. Can be also called that. It exists from prophase, but at metaphase, it will actually attach itself to the chromosomes. Which part of the chromosomes? Specifically the centromere, which is this little central part here where sister chromatids are held together. This is my centromere. These are my spindle fibers, or mitotic spindle. These are, again, the centrioles. The chromosomes then start to be pulled along the two opposite poles of the cell by the spindle fibers. These spindle fibers have the ability to contract and pull each sister chromatid to opposite ends of the cell. And so that's where it starts to prepare them to actually separate. Which then brings us to the next stage, which is anaphase, when the actual physical separation of the sister chromatids is taking place and they're being pulled to the opposite poles of the cell. The separation is aided by motor proteins that are able to contract and physically pull against the chromatids. This is an active process. It requires ATP. So you can sometimes see sort of little visible mitochondria kind of arranging themselves around the spindle fibers so that they can provide ATP. The chromosomes will have this kind of characteristic appearance. The chromatids pulled apart, and they will actually look like they're being dragged through the cell. Beware that many questions will actually show you microscope images of the phases of cell division, and it's a really good idea to sort of train your eye to familiarize yourself. But this will be one of the ways that you'll know you're looking at anaphase versus looking at metaphase, when the chromatids are aligned along the equator of the cell. So each pole of the cell will end up with identical and full sets of chromosomes. That's the whole purpose of first lining up the chromosomes along the equator and then making sure that each daughter cell only receives one sister chromatid each, so that they've got both a full set and an identical set. Since the function of mitosis, part of the function is to create growth or to replace dead or damaged cells, they have to be genetically identical. Otherwise, they wouldn't be replacements of the cell that was lost. Finally, telophase, the nucleus begins to reappear. It's beginning to form again here. So here is my nuclear envelope reforming. And I, of course, have two new nuclei. So, and they're all part in the same cytoplasm. So the cell appears to have two nuclei for this brief moment in time. The chromosomes begin to decondense. So you can see they don't have that distinctive chromosome appearance. It looks like a sort of loose fiber. Decondense. And the spindle fiber also begins to disintegrate. It's not needed anymore. The chromosomes length and uncurl, meaning that they can no longer be seen under a microscope. You may be able to see some kind of diffuse staining, but you won't be able to identify rods. And of course, as we said, the nuclear envelope reappears around the chromosomes, around the chromatin. Let's have a look at an exemplar question here. So after fertilization, a zygote rapidly divides to produce more cells. Each cell cycle may be as short as 30 minutes and only involves DNA replication and mitotic divisions. State the type of cell division involved in this process and suggest why the produced cells become smaller after each cycle. So take a moment to answer that. I'd suggest that you do try to write out an answer, because you can then compare it to the model answer that we're going to give here. And that's going to be really good for you to see exactly where you go wrong. It's going to help you to kind of pinpoint your areas that you need to work on. You could plan the answer in your head, but writing it down is better. Okay. Let's look at the answer together. So the context is the whole first sentence. So the context tells us that we are dealing with cell divisions, rapid cell divisions to produce somatic cells. And so our command is state. So we, in terms of stating the type of cell division, it's just the one, but also suggest an explanation. So that's going to be the rest of the 2 marks. So 1 mark will go for state, 2 marks will go for the explanation. What is my direction? Why each cell division gets smaller. So that is what I need to be able to justify in my suggest part. So this is my direction here. So let's see how we're going to break down this answer. So first of all, let's identify the type of cell division. It's kind of already given to me also in the question here, but it's basically mitosis, since it's somatic cells being produced. So that's 1 mark. I'm just going to briefly state that sometimes this particular type of cell division, where there isn't much growth between cell divisions, it can also sometimes be called a cleavage. It's not in your syllabus explicitly, but just if you see it, that it doesn't throw you off, just have it in the back of your mind, this term. So they divide through mitosis, and usually cells will spend more time in growth 1 and growth 2 phases, where they will undergo protein synthesis and make organelles, which is basically what growth is, isn't it? Making more organelles and synthesizing proteins, translating proteins. Without these, so this is now we are suggesting why these cells are smaller, become smaller after each cell cycle. They spend less time. I was told that because I was told that the cell cycle might be as short as 30 minutes. That was a clue. And it only involves DNA replication and mitotic divisions. So which stage of the cell cycle is missing? Growth 1 and growth 2. Without growth 1 and growth 2, no organelles. Without these stages, the amount of cytoplasm and the number of organelles must be divided between the daughter cells, so they effectively almost half with each cell division, so the size reduces. Let's now move to cytokinesis, which is not a part of mitosis. The cyto means that, like, we're the chapter mitosis and we're looking at cytokinesis, but of course cytokinesis is a separate stage, and it will involve the separation of the rest of the cell components, the cytoplasm and the cell membrane, into 2 identical, of course, because we're talking about mitosis, daughter cells. It happens slightly differently in plants and animals. In animal cells, where there's no cell wall and the plasma membrane is it's elastic, it's non-rigid, it's fluid, right? As the fluid mosaic model tells me. So it will actually fold, and there are 2 dents. Let me just, I hope you can see that. There are 2 little dents here formed. For those of you that want a bit of extra detail, these dents are called the cleavage furrow, but don't feel like you must memorize that, just in case you want to. And it will basically constrict around the whole cell, and it will form 2 separate daughter cells. Plant cells do have a cell wall, which is rigid, it's inflexible. And what will happen, there's not going to be any constriction of the membrane, no folding, there will actually be a gradual formation of cell wall, so cellulose, and then let me just do it in a separate color so that it's obvious, and cell membrane beginning to form. So it's kind of like building a fence, imagine. So I'm going to have plasma membrane, and this structure holds the end plate here, which is made of cellulose. So the end plate will eventually become the new cell wall that separates the two daughter cells. And that will form two separate plant cells. Let's have a look at a typical question, which is also a calculation question. So E. coli can divide as often as once every 20 minutes, as can most bacteria. So that is my context here. Context in green. Assuming that one E. coli cell weighs 1.5 picograms. Quick unit explanation. A picogram is 1,000 times smaller than a nanogram, which is 1,000 times smaller than a microgram. Don't worry too much because I'm actually told the unit conversion here. So calculate the amount of time taken for 24 picograms of E. coli to grow from a single cell. So my context, we said, is the data here. The command word is, of course, to calculate. Do it in red. Calculate. And my direction is what I need to actually calculate here. So take a moment again. I'm going to give you a couple of minutes to see how you would approach that. It's challenging to have a think. Even if you don't solve the whole thing, plan it in your head. So think about, first of all, how you will convert the information into number of cells. How are you going to find the number of cell divisions required since you're only given the amount of time for one cell division? What's the kind of strategy that you want here? Let's break down our approach to this question. So, how much time is taken for 24 picograms of E. coli to grow? First thing I need to do is to convert that into number of cells. Okay, because I'm only being given the amount of time that it takes for one cell to become two, to divide. So I have to convert the information given to me in picograms in terms of numbers of cells so that I can actually work with, so that I can utilize this piece of evidence here, the 20 minutes for one division. So how are we going to convert those picograms into numbers of cells? So I'm just going to write down like our strategy here. Calculate number of cells that are corresponding to 24 picograms. I'm being told that each one cell weighs, this is where you've got to be utilizing every piece of evidence that you're given. So how will I utilize this information that one cell weighs 1.5 picograms? So I will do 24 picograms divided by 1.5, which is the weight of one cell. And that gives me the number 16. Obviously, you know, this is picograms. This is picograms. The units cancel each other out, the top and bottom of the fraction. So that gives me the number 16. So 24 picograms correspond to 16 cells. Okay, now I'm getting somewhere because now I know that I need to go from one cell to 16 cells. So next I need to figure out how many rounds of cell division will this take to go from 1 to 16 cells? How many cell divisions? So well, you can do sort of like sketch that out in your head. So one cell becomes 2, that's one cell division. 2 cells become 4, that's a second cell division. 4 cells become 8, that's a third cell division. And 8 cells become 16, because that's what I worked out here. That's a fourth cell division. So I need 4 divisions to take place. Okay, now the solution is nigh, because I know that each one cell division takes 20 minutes. Just quickly, you can write it out like that if that kind of like makes more sense to you, to count basically the number of cell divisions, or if the numbers were different and it would be impractical to write out all the cell divisions, you could also use the formula 2 to the power, 2 to the power of n. Sorry, that's meant to be an n, not an h. N, where n is the number of cells. Sorry, number of cells is 2 to the power of n, where n is the number of cell divisions. So since I know that each cell division takes 20 minutes, and I know that to go from one cell to 16 cells, I need 4 cell divisions, then my total time, because I need the total time taken is of course going to be 4 divisions times 20 minutes, which is 80 minutes. You can write minutes like that, or just write out minutes just to be sure. Okay,